Hello everyone, my name is Rahul. In the previous video, we had discussed about fragmentation of DNA. In this video, we are going to discuss about amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. Polymerase chain reaction. This technique was developed by Carey Mullis. This technique was developed by Carey Mullis in the year 1983. PCR is a method of amplifying specific regions of DNA molecules without the requirement of time-consuming cloning procedures. So instead of making one clone at a time of a particular gene of interest, we will amplify it using this PCR, and within a very short period of time we will be able to obtain numerous copies of the required gene of the gene of interest. The reaction PCR takes place inside an instrument known as Eppendorf tube. Eppendorf tube. The reaction takes place in the instrument known as Eppendorf tube. So using PCR technique, a very low content of DNA available from samples, very low content of DNA available from samples, or which may be a tissue, which may be blood. It can be amplified many times and it can be analyzed. So this is the second use. We can use this technique for analysis and also for performing the recombinant DNA technology. So for example, from a crime scene, we obtain a small hair cell. So from a small hair, we will be extracting a very little amount of DNA, but we will need to perform numerous tests on this DNA. So to perform these tests, we will need a large amount, we will need more quantity. So what we will do is we will perform the PCR on that DNA and we will be obtaining a very high content of DNA from a small sample. This polymerase chain reaction requires the presence of an enzyme, requires an enzyme called as polymerase. Now this polymerase, the most commonly used polymerase is a Taq polymerase. It is a Taq polymerase, which is a heat stable polymerase. That is, it can perform its function even at very high temperatures. So this Taq polymerase, it is obtained from a bacterium known as Thermus aquaticus. Thermus aquaticus. It is the most commonly used polymerase. Other polymerases that can also be used are, one is known as flu polymerase, which is obtained from Pyrococcus furiosus. And we can also use another polymerase known as Wnt polymerase, which is obtained from Thermococcus littorularis bacterium. So how does PCR work? The main steps in PCR are three. The first step is known as denaturation. The first step is denaturation. In this step, a double-stranded DNA molecule, it is placed at 94 degrees Celsius. It is placed at 94 degrees Celsius. So at 94 degrees Celsius, what happens is these two strands, they will separate out. These strands, they will separate from one another and we will obtain two single-stranded molecules instead of a double-stranded molecule. So at 94 degrees Celsius, denaturation takes place and the two-stranded DNA molecule will be converted into two single-stranded molecules. Now, each of this strand will act as a template. So for now, we will see how the upper strand the upper strand 5 prime to 3 prime, it will act as a template. Similarly, the second strand, it will be also acting as a template. So the next step is annealing or cooling, which is done at 54 degrees Celsius. Annealing or cooling. So what we do is we use a primer which is attached at 3 prime end of single stranded DNA. We use a primer which is attached at the 3 prime end of the DNA. Why it is attached at the 3 prime end? Because through your knowledge on molecular basis of inheritance, you will already be knowing that the polymerase only functions in the direction of 5 prime to 3 prime. So the polymerase that we will be using later on, it can only extend this primer in the direction 5 prime to 3 prime. So we will be attaching this primer at the 3 prime end. The primer will be attached at the 3 prime end. What is the primer? A primer is a small collection of nucleotides, complementary to the template strand. So the nucleotides here, the base pairs will be complementary to the DNA strand, which will be acting as the template. So this primer will be attached here at the 3 prime end. And next step is extension. The extension is performed at 72 degrees Celsius. During extension, we add the DNA polymerase and deoxynucleotides, that is small nucleotides we add. So the polymerase, the tag polymerase which is present, it will extend the primer in the direction 5 prime to 3 prime on this strand and 5 prime to 3 prime on this strand. So both the strands, this strand and this strand, they are acting as template strands, and the primer in the presence of the polymerase will be extended from 5 prime to 3 prime end on both the strands. So from one DNA molecule, we have now obtained two molecules. So in one cycle, the DNA has been amplified. So when 30 cycles will operate, our DNA will be amplified to around 1 billion times. How? The number of times the DNA is amplified will be given as 2 raised to the power n, where n is equal to number of cycles. So in three cycles, 2 raised to the power 3, there will be 8 times amplification. So similarly, in 30 cycles, the amplification is 2 raised to the power 30, which is around 1.07 billion. So from a single molecule, we have now obtained 1 billion molecules of the required gene, of the required DNA of interest. So what are the 3 steps? The 3 steps used are, first is denaturation. The first step is denaturation, where double-stranded DNA at a temperature of 94 degrees Celsius is converted into single-stranded DNA, and both strands will now act as template strands. The next step is annealing, which is done at 72 degrees Celsius. Two primer DNA, that is one for each strand, are attached at the 3 prime end of single stranded DNA. The third step is extension, which is performed at 72 degrees Celsius. Here, tag polymerase in the presence of deoxynucleotides extends the primers in the direction 5 prime to 3 prime. This PCR is an automatic process. Once you have entered all the required compounds, all the required materials in the Eppendorf tube, and you start the reaction will continue by itself. And we have to remember that Taq polymerase, it is a heat resistant enzyme, which is why it is able to act at such a high temperatures of 72 degrees Celsius. So with this, we finish off with this discussion on polymerase chain reaction. See you in next video.